All right. So without further ado, before we dive too far into what the whole concept about SomaLogic is, let's start a bit more fundamentally about why are we looking at proteomics even, right? If we go back to the central dogma of biology, we understand that you have your DNA, it's your genomic material, it makes your RNA, your transcriptomic profile, and then from an RNA it gets translated into proteins, right? So that is the central dogma of biology. And traditionally, DNA and RNAs are really the molecules that we look at from a clinical setting for multiple reasons, and those are most of our diagnostic biomarkers, right? And really, the use of proteins as a diagnostic biomarker has largely lagged behind DNA and RNA, not because proteins are not important, but really because of our ability to characterize them in a robust and scalable manner in the clinic. Traditional antibody affinity methods are limited in their plexing and they face long and unreliable development lead times. If you've been following some of the developments in the literature coming out of Nature, for example, you'll see that there has been a lot of uncertainty and a lot of disagreement even about the field of antibody-based proteomics. So really, if you start thinking about it, the protein oftentimes is the one effector molecule that is driving your clinical phenotype, your biological phenotype that we're seeing within your patient or your study or your sample cohort. And then understanding the interplay between DNA, RNA, and proteins really then helps us layer the information and build a deeper, more cohesive and comprehensive insight into the types of biology that we are studying. So really, what is SomaLogic, right? SomaLogic previously is the leading proteomic company. Now we are the leading proteomic technology platform. Okay, and the one thing that is significant about SomaLogic is that we are not antibody affinity based. Okay, we've been in existence over 25 years, and in that time, we have developed a very robust aptamer based technology that then offers pretty much unlimited plexing for our proteomics assays that would then otherwise limit other technologies like mass spec and antibody-based platforms. We have the broadest coverage in terms of protein measurements with exquisite specificity and reproducibility in a high throughput fashion. And one of the key anchors behind our ability to provide a very robust and high-plex assay really is the use of the aptamer concept as a affinity molecule to then aid in our detection and readout of proteins within the samples that we are assaying. And the reason why this is the case is that the aptamers that we are working with, unlike traditional antibodies, are wholly chemically defined. We are able to reliably, from batch to batch, produce the exact same aptamer molecule, exact same sequence. And we have very in-depth characterization and manufacturing QC steps that then ensures the quality of the reagents that we are manufacturing. All right. We're leading the market in terms of the amount of samples and data that we have output from our assays, okay, with more than 1,200 publications to date on various studies using our assay and our platform. All right. All of these 1,200 papers, we do have multiple journal covers. We also do have landmark assays across Cell, Nature, and Science. And all of this is possible really because our best-in-class robustness and reproducibility of the assay at a median coefficient of variance at 5%, which means that you can discover and understand more biology with less sample and greater resolution across your samples. So what exactly is the aptamer technology that we're using, right? At the very core behind SomaLogic is this thing called a SOMAmer reagent. What we call a SOMAmer is simply a slow off rate modified aptamers, right? So these are just aptamers that we then go in and chemically modify them so that they have chemical side chains that are representative of the kind of chemical functional groups that you will see in an antibody paratope. This then allows our SOMAmer reagent to more or less perfectly recreate the kind of binding affinities that you get with antibodies, okay, but retaining the chemically defined nature of the aptamer. So essentially, with the Soma reagent, you have the best of both worlds. You have the specificity that you get with a traditional antibody, while still retaining a chemically defined molecule that allows you or allows us to robustly reproduce the same molecule over and over and over again, giving us very low percent CV across all of our assays. All right. So the kind of affinities that we get out of the aptamer or Soma molecule is comparable to or even exceeds that of antibodies. All right, and this is only possible because firstly, we start with a very large random library through which we process through our selects process. Okay, and then the fact that aptamers are very small and that we have pretty much unlimited ability to go in and select for sequences that bind to and recognize different proteins really means that we have exquisite specificity with best-in-class scalability as a platform across all of the proteomic technologies on the market today. So one thing about our aptamer or our Soma molecule is that every single Soma molecule goes through an extensive validation process, and we provide data from that validation to anyone who's interested in understanding more about every single one of our Soma affinity molecule reagents. Data from all of these affinity molecule reagents are available on the website at menu.somalogic.com. And I'm going to talk about this later, but there you can see things like the affinity, the limit of detection, the median signal that you would expect, some signal to noise ratio, as well as the kind of proteins that were used as a bait together in the selects process. Right, so we have pretty much the best transparency that you get out of any affinity molecule company on the market today. All of our validation data is disclosed and it is done in a very in-depth manner. So then, what are some of the highlights of the SomaScan assay that utilizes our Soma reagents, right? We have the best-in-class plexing ability with over 11,000 measurements, and these 11,000 measurements covers more than 50% of the human proteome. As part of our assay, we are able to read out on the entire dynamic range of expression that you would expect from the human proteome across multiple different sample types. We do have exquisite binding specificity through a highly validated affinity molecule, all right? And what is wonderful about the SomaLogic technique is that data across plasma and serum samples across multiple different batches can be harmonized for a combined data analysis without the necessity for having data bridging across your different experimental batches. Okay, and all of this is underpinned by a chemically defined molecule that then allows us to give you best-in-class reproducibility and robustness with a median CV of our assay of 5% across human plasma and serum samples. The actual assay itself is highly automated, and this then allows us to rapidly scale and process samples. And the amount of samples that we need is relatively small at 55 microliters of input material in our human plasma and serum assays, for example. So then going a bit deeper into the plex of the assay, what exactly does 11,000 plex mean, right? So you can see that actually the way the SomaScan assay is developed is that it very closely models that of the whole human proteome, right? We have coverage of the secretome, but not only that, we also have broad coverage over membrane-bound targets as well as intracellular targets. And there is a good rationale behind that because what we do see, especially in biofluids, is that we don't just pick up components of the secretome. we are pretty much looking at molecules from all over of the human proteome, whether it's a membrane-bound protein or an intracellular protein. It does make its appearance within a biofluid, whether it's plasma serum, urine, CSF. What you'll see is that you're actually not confined to just looking at secreted proteins. So the wonderful thing also that Plex allows us to do is that we pretty much have more than 50% coverage for nearly all of the CAC pathways of interest that you might want to access to aid in your research, right? And this also offers broad coverage over different biological teams like cardiovascular health, inflammation, oncology, neuro, regardless of the kind of field that you're working with, the discovery team of your research, we do provide coverage of content within that would be relevant to your field of interest. So one of the things that is tricky about the proteins and protein expression really is the wide dynamic range over which proteins are expressing over within different biomolecules, right? And definitely within blood in itself, right? Oftentimes we are looking at more or less a 10 logs of dynamic range or 10 orders of dynamic range. Okay, so we are pretty much the only technology out there that is able to go in and sample your biological samples in their native format, right? Without having to do any form of depletion or changes to your samples, right? And this is possible because we use a 3 dilution strategy where we have a low dilution bin followed by a mid dilution bin and a high dilution bin. And the purpose of these dilution bins really is to ensure that we have optimal coverage of your proteins of interest, okay, and that optimal coverage is then matched to the linear range of detection of our SOMA affinity molecules. So most of your molecules, which are your low expressors, will then come in our low dilution bin, which covers about 85% of the assay content. Another 12% of the assay content comes in our mid dilution bin. Okay, so pretty much the low expressors, the mid expressors, and then the high expressors, things like albumin, or vitronactin, we do dilute it the most at 1 to 20,000 times dilution. Okay, and then this accounts for less than 3% of the assay content. Having this triple dilution strategy allows us to ensure that we are optimizing in terms of protein coverage, as well as making sure that we do not saturate any of the signal and we do not lose any of the signal coming out from any of your mid to low expressors that you would be interested in assaying. Right, and all of these is backed up by an automated assay. Okay, and this automated assay really operates on magnetic beads, right? They're just strep-evidin modified magnetic beads that we then use automated magnetic platforms as well as pipetting to then carry out the assay without the need of human intervention. Okay, so what you'll see throughout this assay process is that we do have a 2-step capture and washing. And these 2-step capture and washing allows us to increase the stringency of the assay to again allow to give you the most robust and reproducible data that you have, right? And this 2-step capture and washing, you'll find it labeled on this diagram here as catch 1 and catch 2. So going very quickly through how the assay is run is that starting out from the factory, we have this thing called catch 0 beads. They're essentially magnetic beads that are already pre-decorated with different SOMA reagents, right? And then these SOMA reagents or magnetic beads are incubated with your patient samples or your samples of interest, from which proteins would then go in and bind to the different SOMA reagents, all right? Well, these proteins are bound to the SOMA reagents, will come in with a biotinylation reagent that will then biotinylate these proteins that are bound to the magnetic beads through the SOMAmer. After which, we'll do a photocleavage and release all of these bound constructs into solution. what we do know about this process is that during the capture process, we will have a mix of specific versus non-specific binding, but that's perfectly fine because at this point, we will have what we call a kinetic proofreading step where mock SOMAmer molecules would then come into the solution, right? And what they'll do is they would off-compete all of your non-specifically bound protein molecules, okay? Such that at the point where we are coming in with our fresh streptavidin modified magnetic beads, okay, and we bind it to it, only proteins that are specifically bound, right, will then carry on their respective SOMA reagent to this, okay? And after that, after a few rounds of washing, we'll come in with a denaturing solution that will then denature the proteins and release the SOMA reagents. So at this point, you've heard about Illumina, all right? What is nice about the SOMA reagent is that it's really amenable to multiple different methods of readout. We can just take this SOMA reagent, directly splay it on an array, and from there read out on fluorescence. Or if we really want to, we can also use an NGS-based assay to then read out on the type of protein targets that are coming out from our sample. Essentially, the entire upstream process of the assay is consistent and the same, and all that differs is how we are counting the SOMAs that are within our eluted material. So as part of the development process of the SOMAs, okay, each SOMA does undergo a very stringent and comprehensive development and assay validation process, right? And this then goes on to establish the sensitivity, binding affinity of the SOMA, right? Screening against non-specific high abundance proteins to ensure that, we do not have non-specific stickiness, for example, to albumin. And as a result, because there's so much albumin, I get carryover into my actual signal. Okay, and as part of that, we actually do a conformational binding pull-down assay using both Western as well as mass spec conformation to make sure that when we, that we are exactly getting the protein of interest that we want, okay, when we isolate these proteins from samples, right? And from there, we do estimate things like the plasma concentration of the cognate protein, the dilution and linearity ranges that we do have for the cognate protein as well as the corresponding SOMA reagent. And from there, what then is the optimal assay dilution that we're looking at? Once candidate SOMA reagents have been developed, we actually put them then through an assay type validation where SOMA reagents are mixed in with the broader SomaScan assay, okay? And then we'll look at establishing things like the coefficient of variance of each individual SOMA reagent, the precision, okay? And then we also look at establishing what the data looks like against our population reference, which is our Covens reference population. Other things that we'll get would include the signal to noise ratio, the LOD, as well as population F statistics and any form of correlation that might come up from the assay. In addition to that, about 80% of the assay has additional third-party orthogonal conformation, and this will come in the form of mass spec validations from third-party literature sources, our in-house pull-down assays, as well as cis-PQTLs or any form of publication that does mention the SOMA reagent as well as the target of interest. So how this process looks, as I mentioned, we first start with a selects process, and selects just stands for a selective enrichment of ligands through exponential enrichment. What this is it's starting with a randomized library of 10 to the 15 different SOMA sequences. We then allow these SOMA sequences to interact with our protein bait, okay? And these protein bait would just be your proteins of interest, right? And from there, we do recover specifically bound SOMA reagents pretty much in the same fashion that we perform our SomaScan assay. We do this multiple times, and over the process of doing this enrichment and washing, we are increasing the stringency of binding, all right? And this then allows us to get exponential enrichment of our selected sequences. From there, some of the data characterization that we get include a dilution test using our diluent. Okay, we also do have a spike in dose response, right, where we spike in the actual protein target into our different matrices of choice, for example, plasma, as well as blank buffer. Okay, and then we get things like the dynamic range of signaling of each individual protein and SOMA reagent. So as part of validation, once each SOMA reagent is completed, I did mention again about the putting each SOMA reagent in the context of the SomaScan assay. And this is going more into detail about some of the characterization information that you'll get out of every single SOMA reagent that you'll see on our menu.somalogic.com website too, right? And after this session, please feel free to play around with that too. It will require you to register for an account, but once you register for an account, feel free to click around, check out your proteins of interest, check out the types of signal that you get. For example, the median, the 95 percentile signal that you get, as well as things like the LOD, population F statistics, that really helps you start thinking about and building up the kind of science and kind of biomarkers that you might be interested in within your specific models of health or biology. So I mentioned in terms of orthogonal conformation, about 80% of our menu or our total SOMA reagent, right, has one or more forms of orthogonal conformation, right? And then these orthogonal conformations can come in the form of, for example, small or midplex antibody techniques like ELISA, or other forms of antibody panels, mass spec conformation, right? And then in-house, we are also working on the confirmed absence of binding to nearest neighbors. So depending, especially when you come into broad families of proteins of interest like tyrosine kinases, right? The confirmed absence of binding to a very similar type of protein construct actually is a very big part of ensuring the robustness and validity of your data, right? Mass spec from literature, we also do mass spec in-house, right? And then correlation of mRNA, this would cut across human cell lines, cis-pQTLs, okay? And all of these information on orthogonal conformation is also available on our website, where we first go through a selection process where we have different oligos in a randomized library binding into our protein baits of interest. We recover specifically bound sequences, and then we put them through a cyclic process to then achieve the exponential enrichment in selects, right? As part of this process, we get a lot of validation data that comes out of each individual molecule that includes a dilution of plasma, right? A dose response curve when we do a spiking of the specific protein, as well as then the dynamic range of response for each specific affinity molecule of interest. Okay, and then as part of that, we also do a mass spec pull down, as well as a Western conformation that we're actually getting binding to our specific protein targets, right? So part of this process, we actually do get multiple reagents against a single protein of interest. So one of the things that we also do would also then be affinity selection and tuning to make sure that we get the optimal reagent included within our actual assay itself out of all of the candidate SOMAs that come up for a specific protein. All right. Each of these candidate SOMAs are then placed into the context of the SomaScan assay, where we'll look at things like QC replicates. From there, we get a lot of the matrices on things like percent CV, stability of the assay, F statistic, as well as, very importantly, establish a population reference data point for all of our protein measurements, right? And I did mention a bit about the harmonization of the data. What allows data harmonization really is the fact that we have our own internal population reference control, that we can then use to serve as a benchmark or baseline for comparison across multiple different assays. Okay, I think this is pretty much where I left off, where I was talking about the different forms of orthogonal conformation. Roughly about 75 to 80% of our SOMA reagents have one or more forms of orthogonal conformation. Okay, you can see that, you know, a good amount, pretty much half of the 8,100 SOMA reagents have two or more different methods of validation. And then this can come in the form of small or mid-plex antibody techniques, various forms of mass spec conformation, whether it's a pull-down assay, it's a buffer conformation assay, or whether it's just a literature published mass spec study where they did their own conformation of the SOMA reagent that they use in their study, right? In addition to that, like I mentioned, we do have an increasing library where we are looking at confirmed absence of binding to nearest neighbors, okay, as well as correlation with mRNA across cell lines and cis-pQTLs, right? And all of this information, like I mentioned, is available at menu.somalogic.com. Right. So going a bit more into the menu to menu.somalogic.com, all right? When you after you registered for an account on the website and you sign into it, you can click into the different proteins of interest, right? You just look for example, E3 ubiquitin protein ligase. Otherwise, you can also search for different accession IDs, like the target name, the seq ID, or a UniProt ID, or even an on-trace gene ID, right? From there, it will tell you, for example, how many different SOMAs we have targeting a given target of interest. We also tell you the amino acid range that was used to bait the selects process. For example, in terms of ZNRF3, we have two different SOMA reagents, one which is a full-length protein construct that was used to bait it, and then the second SOMA reagent was only developed against amino acid range 56 to 219, right? So this is significant because different parts of a protein does read out on different types of biology, right? And for example, we do see this becomes very significant in the case of selective molecules, for example, collagen 6A3, right, where there is a difference between the C-terminal and the N-terminal biology of this particular protein target. So in addition to that, I did mention a lot of confirmation and validation data. As you scroll down in the page, it will start telling you things like any form of secondary annotation, orthogonal conformation, as well as binding to nearest neighbor, right? And then this is where we'll see things like no binding was observed, you know, in two different types of nearest neighbors or things like that, as well as the performance of the particular affinity molecule within the context of human serum or plasma. Right. So this really allows you to start thinking about the molecules, biomarkers that you are interested in, understanding the types of coverage we have for that particular series of biomarkers. And from there, the kinds of signal that you can expect out of your samples that you'll be running on our assay, right? And again, all of this information is available for every single one of our SOMA reagent, right? And this is only possible because we are upfront and transparent about the validation process, and we release all of that information that we get out of the development and validation process of each SOMA reagent into the menu.somalogic.com tool. So then, what kind of data are you getting out of the SomaScan assay, right? The data coming out of SomaScan is actually just a simple count matrix, right? And a simple count matrix actually is fantastic because it means that your downstream data manipulation becomes relatively straightforward. All you need to worry about then is the biology, and you don't have to worry about, you know, some of the more convoluted file types that can come out of high-plex data analysis, right? So for the data that we deliver as part of our assay, right? We deliver two different forms of count matrices. One that we call ANML normalize, ANML for adaptive normalization by maximum likelihood. And then one is pre-ANML normalized that we just call pre-normalization ADAP. All right. So the ANML normalization is essentially just pinning the data that you get from every single sample against our reference, internal reference covens population. Okay. And this internal referencing is what really allows us to then bridge data across multiple different experimental batches, across longitudinal or latitudinal studies, without having to worry about things like data bridging, sample bridging, right? All of that data is then you can just batch analyze all of your data without having to worry about experimental batching. Okay, so going a bit deeper into the types of data manipulation process that we do across our SomaScan data type is that for the data that comes out of the SomaScan assay, we first perform a hybridization normalization where we look at height pause controls, and essentially what we are adjusting for is hybridization efficiency, in this case to the sample as well as to the array slide itself. From there, we will do an intraplate median signal tweak to ensure that all of the data that's coming out from the SomaScan assay itself is placed on a relatively similar baseline, okay? And then we'll do plate scaling as well as calibration of individual Soma reagents, right? This is only possible because we do run calibrating samples across every single one of our assay runs, all right? And these calibration samples are then again referenced against our calibration, our master calibration mix, so that we know exactly how to go in and adjust for differences in hybridization or binding differences for any single Soma reagent. So what you can see is that, in terms of how these steps affect the data, right? So these are data taken from 3 representative individuals, okay, and then done across multiple different plates of analysis. You can see that the raw data itself, well, we do see some form of grouping of the data, some clustering, but that clustering can be relatively broad. As we go down the steps of height normalization and plate scaling, okay, the data starts getting sharper and sharper in terms of clustering into the individual patients, okay, and to the point where after calibration, right, we can more or less identify 3 distinct clusters for the 3 different patients, right? After which we do pin the data from these 3 different individuals against our covens reference population through ANML. And what this then allows us to do is to make sure that when we analyze the data, we are looking at it from the same reference baseline without having to worry about things like bulk expression, fold change differences across different samples. So a different view again of what the data looks like as we go across different data standardization and normalization process, okay? And you can see that after ANML normalization, we can really start digging into differences in the biology, right? So one of the big questions is that as we go about increasing our plex, right, does it really affect the assay specificity, the robustness of the assay? And the short answer is no, okay? And this is only possible, again, because we're using an aptamer molecule that is really small. So we don't have to worry about things like steric hindrance or crowding within a bulk solution. Therefore, we really don't have to worry about a maximum upper limit in terms of the amount of plex that we can get at. Right, and you can see this in terms of the total CV, the median total CV that is preserved across all of our assay plex, whether it's 5K, 7K, or 11K. Right. So there are a bit of changes in terms of to our signal to noise ratio, but it's still relatively stable with more than 99% of the content signaling above a signal-to-noise ratio of 2. Okay, and then same with the sensitivity in buffer. So once you get your data, there are multiple ways that you can analyze your data, either through our online stats.somalogic.com or through our GitHub repository, where we deposit the code needed to analyze your data, right? And whether you access it through our GitHub or through our online tool, you're able to get access to the same full suite of statistical tests as well as visualization tools. Right? And these statistical tests can be as simple as correlation, or it can include different statistical models like t-test, u-test, ANOVA, okay, as well as allow you to go in and plot different volcano plots. All right, so I'm rapidly approaching the end, okay, and I just want to go over very quickly what exactly is available for you as a potential user of the SomaScan assay. We have the assay itself, which are sold in either the 7K or 11K panels. Okay, and then these panels do have sub-panels that you can potentially select for. All right, if you're interested in zooming in on different specific areas of biology that are thematically useful for your study, right? So in addition to this, one of the key features of the SomaSignal test, one of the key features of the SomaScan assay really is this thing called the SomaSignal test, right? So what the SomaSignal test is that they are biomarker models that we have trained out of our different data sets, okay? And then these biomarker models would then tell you about things like cardiovascular risk, lung cancer risk, NASH or MASH status, as well as general wellness like kidney health, metabolic health, muscle mass, right? So good stuff like that, right? And this is very important because we're starting to go into more about the clinical utility of a high-plex discovery tool that has the best precision available on the market to date, right? It's because of our precision that we are then able to go in and establish different clinically relevant models. Do note that these models are currently administered under a RUO label, which means that, for example, in our US lab, they are administered under a LDT use case in our own in-house CLIA and CAP accredited lab. All right. As part of that, aside from the wide range of biomarker models that read out on different aspects of disease and health, there's also this thing that might be interest called pre-analytical variation. And essentially what it allows you to do is to track different batch effects that might be affecting your data. And these batch effects can include differences in sample collection, differences in sample storage, right, freeze thaws, or even transportation. And this then also allows you to have a larger confidence in your data, to know that, when you're reading out on whatever data, whatever biomarker model that you're trying to develop, that your data or your model is not being confounded by things like sample-to-sample variability due to processing, okay? So again, a different look of the current series of validated SomaSignal tests that are available to access. Do note that the SomaSignal test will require the base SomaScan assay to be performed first. And from there, you have the option of accessing an add-on of a SomaSignal test. All right. So I'm just going to jump over very quick and tell you, the SomaScan assay itself, it's largely developed on plasma, serum, as well as CSF. But at the same time, we also have performed pretty much the same series of assays on any kind of animal model you can think of, whether it's hamster, rat, dog, mouse, non-human primates. or any kind of biofluid of interest. For example, nasal lavage samples, wound fluid, exosomes, right? Any form of aqueous vitreous humors, right? Anything, any kind of biofluid, essentially that contains proteins, we can go in and analyze it on our assay, right? So very quickly, I've been talking a lot. I just want to end off with a quick demonstration, of the clinical utility of the SomaScan assay. And this particular work was done by Novo Nordisk itself, all right, as part of the developmental process when they're rolling out semaglutide. Okay, they essentially had two different cohorts. Step one, which was a pre-diabetic cohort, and then step two, a diabetic cohort that they then treated across 68 weeks with semaglutide treatment with different dosing. All right. All of these patients will follow up with our 7K SomaScan assay. All right. And I'm just going to jump very quickly. And essentially, when we start looking at the baseline data of these patients, first and foremost, high-level sanity check, do we see an increase in cardiovascular risk with the number of comorbidities that are present within the patient? Yes, we do see an increase in the number of in the cardiovascular risk together with comorbidities. Secondly, after adjusting for the effects of weight loss, right, and BMI, when on treatment, we do see a very strong cardioprotective effect coming out from the different semaglutide treatment groups, right? And do note that this is in the space of 68 weeks, okay, where we haven't had sufficient time to go in and actually test for cardiovascular risk events, right? We're just purely reading out on the risk score of these patients, okay? And very nicely over the course of a much longer clinical trial, the cardiovascular effect also did come out, right, from Novo Nordisk's own clinical trial data sets. All right. So in addition to this cardiovascular risk factor that was done using our cardiovascular risk SomaScan test, essentially what you can do is also go in, annotate all of your proteins via their CAC pathways of interest, and we start doing pathway scoring, right, to do more or less a systems biology approach in terms of the data. And we do see broad effects when on semaglutide treatment compared to a naive population like the decode population, okay, and all of these broad pathways that are perturbated when on semaglutide treatment, right, do have associations then with obesity as well as diabetes. Okay, so I'm just going to jump over very quickly next to talking more a bit about a biomarker discovery process, right? I talked more about applying SomaScan test in a more clinical relevant research setting. But in addition to that, you can actually go in and pen for biomarker models that might be of interest to you. So in particular, this is an organ aging study that was done out of Stanford, okay, out of the Tony Wise group. And what they did essentially was take the entirety of the SomaScan assay, okay, and start annotating different protein targets according to the putative organ of origin, all right? And they'll look at things like, other clinical measurements, like things like blood pressure, pulse, CRP, blood count, right? Very extensive clinical metadata data set. Okay. And what they did out of this was they started establishing a model of what an organ would look like in terms of the proteomic signature during the process of healthy aging. Okay, and this has been validated across multiple different study cohorts, our own internal Covens data set, right, in a longevity study cohort, right, the SAMS brain MRI data set, the Stanford ADRC, as well as the Knight ADRC cohorts. So when we start looking at the utility of organ aging or trying to then stage, what an organ health looks like with regards to biological age, okay? if we just zoom in, for example, on cardiovascular risk, and we look at the hazards ratio, right? What we do see, for example, in terms of cardiovascular risk factor as read out from our cardiovascular disease SST, there is a very strong correlation to heart aging, all right, as well as secondarily things like kidney, adipose, muscle, brain, okay? And if we start looking then at total mortality, okay, and then again doing the same kind of analysis, very strongly we do see things like just a conventional aging, orgasmal aging, as well as heart and adipose coming out as some of the top factors that do affect total mortality risk as well as cardiovascular risk. So this becomes really interesting because we're starting to build different models of biology that we can then validate against clinical data, right? And this is more of a discovery process. We're going in with a super high-plex panel and panning for biomarkers that would then be relevant to our model systems, organ systems of interest, right? So that's pretty much it. I'm just going to leave off again, emphasizing that we are pretty much the highest plex game in town. We are the plex leaders, offering more than 50% of coverage across the human proteome. A lot of the studies that you see are only possible because we are the plex leaders, right? All of this is offered against an unparalleled 10 logs of dynamic range, going from femtomolar to micromolar, okay? And exquisite specificity because of our chemically defined affinity molecule, the SomaMer itself, together with the ability to harmonize data without having to worry about sample bridging across different experimental batches, the highest reproducibility that you get out of any assay on the market that's commercially available, right? All in using a very small footprint of sample, 55 microliters of sample.